Homeostasis is the maintenance of a stable internal environment in the body. It keeps things like water, pH, small molecules, and ions within specific ranges required for normal cellular function. This maintenance is achieved through exchange across the cell membrane and through various systems designed to maintain ranges both within cells and throughout the organism. The human body undergoes complex interactions to maintain homeostasis by ensuring systems function to keep different variables within their normal ranges. This is essential for survival, and an inability to maintain homeostasis can lead to diseases like diabetes, dehydration, hypothermia, and allergic reactions. An example of homeostatic control are things such as body temperature, blood pressure, blood glucose levels, etc. All of these systems rely on several components, starting with a receptor, such as temperature receptors in the skin and brain that can detect a change in a variable, which is the stimulus. For temperature, it would be the change in temperature. This information is then communicated through afferent pathways to a control center. Your afferent pathways are usually blood or neurons, and your control center is usually the brain. The brain then has the set point which our body is trying to stick around. After the control center decides what to do with the stimulus, it sends the response through efferent pathways, which are once again neurons or blood, which then go to effectors, and those effectors can then change what is happening and to keep us within that set point. So if it was body temperature, it would be sweating if we were too hot and shivering if we were too cold. Homeostatic control systems are often operate through feedback loops. So there's negative and there's positive feedback loops. So to start with negative feedback loops, it occurs when the effector's response counteracts the initial stimulus, bringing the variable back towards the set point. For instance, if the body temperature increases to 99 degrees Fahrenheit, the sweating response acts to decrease it back to the 98.6, which is the set point, an effect opposite of the initial change. Other examples of negative feedback loops include the replenishment of oxygen by the lungs, the regulation of blood pH at 7.4, and the regulation of blood glucose by insulin. The set point for a variable can sometimes be temporarily and beneficially changed, such as the increase in body temperature during a fever, to aid in the immune system, and this is still considered a negative feedback response because the body is actively working to maintain the new higher set point. In contrast, a positive feedback loop occurs when the response to a stimulus results in a change that the variable increases the deviation from set point. Most positive feedback loops arise when negative feedback systems fail to adequately correct a problem. An example is the drop in blood pressure due to significant blood loss. The negative feedback response of increased heart rate might be insufficient to restore blood pressure. This can lead to less blood flow to the heart, weakening contractions and further lowering the pressure, creating a cycle of increasing deviation from the set point. Such situations often require medical intervention. There are some systems also employ feed-forward control, which is an anticipatory response that occurs before a change in the variable is actually detected. For example, the salivation response to the sight or smell of food prepares the digestive system for when food is finally eaten. Feedback loops rarely operate in isolation and are a part of a larger networks with complex interplays because nothing in our body system works on its own. There is also a hierarchy among these loops, with maintenance of brain having a huge priority over other systems. Other important themes in homeostasis that there's tons of redundancy, multiple systems have crucial variables, and we're able to acclimatize to different things based on where we are and what we are doing. One other thing that we're going to talk about today is protein-mediated transport. The three are diffusion, carrier-mediated diffusion, and active transport, which can then be divided further into primary and secondary. We're going to start with regular diffusion. So diffusion through channels. A couple things about it. Their specificity. Protein channels are generally very specific, allowing only certain ions or small polar molecules to pass through. For example, a K+ channel specifically facilitates the movement of potassium ions. There's different rates. Diffusion through channels is typically rapid when the channel is open because it relies solely on concentration gradients or electrochemical gradients of the substance. The direction of movement is always down the concentration gradient, from a high concentration to a low concentration. As for energy sources that we need for diffusion through channels, this is a passive transport process requiring no cellular energy input. The driving force is the substance's concentration gradient, so from high to low once again. When it comes to competition can occur if multiple substances have similar size and charge and can fit into the same channel, potentially reducing the rate of transport for each of them. The next thing is saturation. Channel-mediated diffusion can exhibit saturation if all available channels are open and the rate of a substance availability exceeds the transport capacity. For the last thing is regulation. Many channels are gated, meaning their opening and closing are regulated by specific stimuli. These stimuli can be electrical changes, mechanical stimulation, or chemical binding. These things are voltage-gated channels, mechanical-gated channels, and ligand-gated channels. And that's all we have for diffusion through channels. To go back to our original chart of all protein-mediated transfer, we're going to go to carrier-mediated diffusion. Carrier-mediated diffusion, let's start with specificity again. Carrier proteins are specific for the molecules they transport, similar to enzymes binding to their specific substrates. The next thing we're going to talk about is rates. Carrier-mediated diffusion is slower than diffusion through open channels because it involves the physical binding of the transported substance to the carrier protein, a conformational change in the protein, and the release of the substance on the other side of the membrane. The rate of transport is limited by the number of available carrier proteins and the speed of their conformational changes. This is once again a passive transport process, which means we know we do not need ATP or any type of cellular energy to get this accomplished. When it comes to competition can occur if structurally similar molecules compete for the binding site on the carrier proteins. The presence of one molecule can reduce the transport rate of another if they both rely on the same carrier. Carrier-mediated diffusion can be saturated when all the carrier proteins are bound to the transported substance and the transport rate reaches its maximum value. It further increases the concentration of the transported substance will not increase because all of the carrier proteins are bound and proceeding through the membrane. Direction is always down the concentration gradient, always from high to low. Regulation, the number of activity in carrier proteins in the membrane can be regulated by cellular signals to either go, stop, come in, don't. That's all we have for carrier-mediated diffusion. Now moving on to active transport, we're going to start with primary. Okay, this type of transport directly uses ATP to move substances against their concentration gradients. Specifically, their specificity, primary active transports are specific for the ions or molecules they are moving. A great example is the sodium-potassium ATPase pump. It is super helpful in most of our cells, if not all of our cells, to get those concentration gradients how we need them. The rates depend on the availability of how much ATP we have and the number of functional pumps that we are using. Energy sources, like I said before, we always use ATP, at least for our NAK pumps. It can also be GTP or any other cellular energy. It's just key to know that energy is being used. For competition, there's not usually direct competition when we talk about primary active transport. While there are a few things that can be interpreted, not always true. As for saturation, primary active transport can be saturated when the pump proteins are actively transporting at their maximum rate. Direction is against the concentration gradients. We're going to go from low to high, which is opposite to our other transports that we've talked about so far. For regulation, the activity of primary active transporters can be regulated by various intercellular signals or hormonal influences. And this is active transport primary. The last type of transport that we have to talk about is active transport secondary. To start off, active transport secondary, this indirectly uses energy stored in electrochemical gradients that are created by primary active transport. Secondary active transport doesn't happen if primary active transport hasn't already occurred. We need that electrochemical gradient. Specificity, secondary active transports are specific to the co-transported or counter-transported molecules from the primary active transport. Directions, there's two directions to go. We can have symport or antiport. Symport move both substances in the same direction, while antiport is going to move them in opposite directions. When it comes to rate, the rate of transport depends on the steepness of the electrochemical gradient of the driving ion. Most of the time it's sodium, but it can be other things depending on the cell that we're looking at. So it depends on that and the availability of the transporter proteins. Energy sources, the energy source is indirectly coming from your primary active transport, such as the electrochemical gradients created by your sodium-potassium pumps. For competition can occur between structurally similar molecules for either the driving ion binding site or the site for the substance being moved against the gradient. Saturation is going to occur very similar to one of our other ones we talked about. It's that when all the transporter proteins are already operating at their max. Direction, one substance moves down its electrochemical gradient. Like said before, regulation, the activity of the secondary active transports can be influenced by factors affecting the primary active transport that establishes the driving ion gradient by local regulatory signals. The last thing we're going to talk about is osmosis, osmolarity, and tonicity. Osmosis is defined as the diffusion of water through a selectively permeable membrane from an area of high water potential to an area of low water potential. For osmosis to occur, the membrane must be permeable to water, but impermeable to the solute, and there must be a difference in the solute concentration on the two sides of the membrane. Water moves through special protein water channels called aquaporins. Osmosis is a passive process. It requires no cellular energy. Water will continue to move until solute concentrations are equal, or until an opposing force, such as osmotic pressure, prevents further water movement. Osmotic pressure is the minimum pressure needed to be applied to a solution to prevent the inward flow of water across a semi-permeable membrane. Osmolarity is the total concentration of solute particles in a solution. It is a measure of the number of osmoles of solute per liters of solution and is typically expressed as osmoles per liter or milliosmoles per liter. Osmolarity reflects the total number of solute particles regardless of their chemical nature or size. On the other hand, we've learned molarity in other classes and in this class. And that is the measure of the concentration of a specific solute in a solution and is expressed as the number of moles of that solute per liter. Molarity focuses on the number of molecules of a particular substance, while osmolarity focuses on the total number of osmotically active particles, which can include ions and molecules. For substances that dissociate in solutions, such as sodium chloride will dissociate into sodium and chloride, their osmolarity will be higher than their molarity because 1 mole of substance produces more than 1 mole osmotic properties. For an example, 1 mole solution of NaCl has an osmolarity of 2. Now, what's the difference between osmolarity and tonicity? They're related, but they're different in very important ways. Osmolarity describes the concentration of solutes in a solution compared to another solution, without considering whether the solutes can cross the cell membrane. Tonicity describes the effect of a solution on how the cell can adapt once placed in that solution. Tonicity is determined by the concentration of non-permeable solutes in the solution relative to the cell. A solution can be isoosmotic to a cell, meaning that it has the same osmolarity as the intracellular fluid, but it can still be hypotonic to the cell, causing the cell to swell. If it was hypertonic, it would cause the swell to shrivel. This occurs when an isoosmotic solution contains permeable solutes that can cross the cell membrane. If a cell is placed in an isoosmotic solution containing a permeable solute, the solute will move down its concentration gradient into the cell. The increase of permeable solute will increase the total solute concentration inside the cell, making it hyperosmotic relative to the extracellular fluid. To restore osmotic equilibrium, water will then move into the cell, causing it to swell. It can also happen the other way. It happens with 5% dextrose. If you inject that into a body system, the dextrose can cross into the cell, but once it's in the cell, it gets metabolized and disappears, leaving us hypotonic. Most of the time for IVs or other hospital needs, we have 5% dextrose and 0.9% saline. I thought it was important to include that those are the main two that we'll use. Then there's a bunch of different combinations of the two that are used for different circumstances. If we want a specific example, we can say a 5% dextrose solution is isoosmotic with any body fluid. However, dextrose is permeable to cell membranes in any healthy individual. And once inside the cell, it's metabolized and will effectively disappear. It's this effect, this continuous removal of intracellular dextrose maintains the gradient for glucose to enter the cell, continuously increasing the intracellular solute concentration. Consequently, water moves into the cell by osmosis.